All right, thank you so much, guys, for coming today. We're going to do our second last lecture today, 18. We'll talk about meninges, which are three layers of membranes that cover and protect the brain. And then we're going to talk about cerebrospinal fluid, which fills the ventricles in the brain and also cushions and protects the spinal cord and the brain from the cranium. So here goes. When I was looking at your LearnSmart modules, I think one of the troubles you had was listing the structures from superficial to deep, including the membranes in their order, all right, and the spaces between those and what's found in those spaces. So we'll kind of sort of go through it quite a bit. So the meninges, we've seen them before when we talked about the spinal cord, right? We know about them already. We know about the dura mater, which we said is a hard, very tough, fibrous layer, and it's tough. That's the dura mater. The arachnoid, we said it looks like a spider's web. That's why it's called arachnoid, from the arachnids. Is the middle one, and then the pia mater. Pia, it means affectionate, and it's most intimately connected to the brain. So that's where that is. And it's rich in blood vessels. All right. The thing about the dura mater in the brain is that it is made up of two layers. Because the cranium, the flat bones that surround the brain, they have a membrane that we've seen before, the periosteum, right? So the periosteum of the bones, okay, which is the membrane, the outer membrane that covers bones, is actually what forms the first layer of the dura mater in the skull. And that layer is called the periosteal layer. And this is from the periosteum, okay? The outside membrane covering the bone. So that's layer number one, okay? And then the inside layer of the dura mater, okay, is the actual meningeal layer. Okay? So I guess in the exam, that's what you had trouble with, starting with the periosteal layer, and then you get the meningeal layer. It is the meningeal layer that is continuous with the dura mater DM of the spinal cord. All right? Spinal cord. So the meningeal layer is continuous with the dura mater in the spinal cord. All right? So remember when we talked about the brain structure last time, we talked about how the brain has got major fissures that we were calling cracks affectionately last time. Okay? Major fissures, especially like the one in the center, which is the sagittal plane, okay, on the sagittal plane. So that's where you have the longitudinal fissure, okay? And that will separate the left and right hemispheres. So what the dura mater does is that it will fold into those crevices, separating those parts. And those parts of the dura mater that fold inwards into those fissures are called dural folds, right? And the part where you have the dural folds, okay, because the dural fold will fold into the brain tissue, separating them, then the periosteal layer continues on the top, right? On those parts, you get to create spaces that are called dural venous sinuses. Okay? Do you see now? Do you see how we form the dural folds and how we form the dural venous sinuses? So let's say this is the skull, okay? And then you have your periosteum like that, right? That's the periosteal layer. And then you have the meningeal layer coming down like that, and then it will curve inwards like that, right? And then this one will do that, and then we'll do that. So this space here is what we are calling the dural venous sinus. Okay? And blood flows in there. All the blood that is serving and supplying the brain will drain into the dural venous sinuses, right? And at the bottom, there's another one, okay, like that. And it's also called the dural venous sinus. So the top one is called the sagittal venous sinus, the superior sagittal, from the sagittal plane, venous sinus, all right? And then the one on the bottom will be the inferior sagittal venous sinus, right? Okay, those two. So you should be able to tell me how the dural folds are formed and what is flowing inside in the dural venous sinuses and what is flowing inside the sinuses. All right. The sinuses, there is blood in it. All right. All the blood that's supplied to the brain is found in there. And also, when we start to talk about cerebrospinal fluid, towards the end of the lecture, we'll talk about how all the cerebrospinal fluid will circulate in all the ventricles, all the 4 ventricles, and the central canal of the spinal cord, and then it will come and it be dumped into the dural venous sinuses. Okay. The last place that the cerebrospinal fluid goes to is the subarachnoid space, kind of surrounding the brain, and then there will be sort of extensions called granulations. That will go into the dura mater and deposit into these dural venous sinuses. So the cerebrospinal fluid is circulated. You continuously make it, and then it gets, turned to the venous circulation, and then you make some new cerebrospinal fluid. All right. Okay. So we know that the arachnoid is this middle. Meninge, right? And then underneath it is the subarachnoid space. That's where you find the, cerebrospinal fluid. Okay? All right. So we got our bone basically on the top, the periosteal layer of the meninge, and then you got the meningeal layer of the dura mater. Then you have the arachnoid. Then under the arachnoid is the subarachnoid space with the cerebrospinal fluid. Okay? And then under that with the pia mater, then brain tissue. Okay? So this cerebrospinal fluid helps to cushion the brain from the cranium. Alright? The pia mater is one that is directly connected to the brain, and it's it's also blood vessels, all right? And when we dissect through the brain, you'll probably be able to see the dura mater, but you won't be able to see the arachnoid. You might see some a little something that the pia mater probably won't, because it's intimately connected to the brain. All right? Okay, so that's that. So that'll be able to explain to me, how those dural folds are formed, what's contained in them. Those dural venous sinuses. So the meninge on the outside, and of course the cerebral or the longitudinal fissure, if you will. That's where the dural fold will, the sagittal dural fold runs into. Right, so this is just looking at it from the other side. All right. This is what you see before you remove the dura mater. So the brains we're going to get for the sheep brains, they will be covered with the dura mater. It's very tough, okay? Especially the dura mater that separates the cerebellum from the cerebrum. That's extra tough. You'll find it hard to even cut with your scissors when we dissect through the brain. Because it kind of cradles. See, the brain is kind of sort of a mushy tissue, so it's kind of cradled, just almost like a bra does. The breast, and it cradles the the cerebrum and also the cerebellum. So it's extra tough. All right. So and that's called the gentorium cerebellum. Right. And this one shows those dural folds. Okay, those special dural folds. Okay. So the folds cerebelli, This one separates the cerebellar hemispheres. Okay. Sometimes I ask about these dural folds in the exam and ask you to tell me, okay, which dural folds separates which structures in the brain. So the folds cerebrally separates the cerebellar hemispheres. Right? So that's the folds cerebrally from cerebellum. Right? And then this one, you would guess what it separates, right? Folds cerebri. It separates which one now? Huh? Separates what? What does it separate? Which? This dural fold that runs on the sagittal plane. Separates the cerebral, the main hemispheres, cerebral hemispheres. You see, cerebri from cerebral, cerebelli from cerebellum, right? You see that? So separates the cerebral hemispheres. This is my shortcut for hemisphere. Okay. That's the folks cerebri from cerebral. Okay. And this is what I was talking about, that cradles the cerebral hemispheres from the cerebellar hemispheres. Okay. This is extra tough. It's actually thick to cut through. You know, you find it very difficult when you dissect the brain. So this one separates what and what. I just told you the two things. Tentorium cerebelli separates what and what? Huh? I just said it. Seriously. I just said it, guys. Is it too cold for you? The temperature is too low. the cerebrum from the cerebellum. Okay, separates cerebrum from the cerebellum. Remember, what did I say cerebellum means? What did I say cerebellum means? Cerebellum? Cerebellum, what does it sound like even? It sounds like a girl's name too. It's small. It's a small brain. It literally means small brain. Because it's shaped the exact same way as the cerebral hemispheres. Okay, it's got two cerebral hemispheres connected by the vermis, okay, which we said looks like a worm. We said the cerebral hemispheres, the main parts of the brain, are connected by the corpus callosum, right? Remember? So cerebellum literally means small brain. So that's why this membrane is extra tough. I mean, this dura mater, which is a membrane, of course. But you know, it's really tough, and you will see for yourself. And this is just showing you the lateral view of the cerebrum. So those are the three main dural folds that separates these major parts of the brain. Okay, and I want you to know all three of them and their name and what they separate for the exam, because I normally bring it up. You can't find it. It should be there. It's playing. It's all over the place. The folks cerebrum, it's not in there. Okay. Anyway, I gave them to you. But it should be in there. You know? Yeah, those are them though. Because those pictures actually separate artwork that I get from the book. No. These pictures, they're extra artwork that comes with the material that I get from the website, which is from this book. For some reason, I'm not sure. Yeah, the folks cerebrum, they should be in there. I'll try to look for it later on and see if I can show it. But yeah, they should be in there. You see, they're all over the place. You see here, this is showing an actual MRI of an actual human being. Okay, the coronal section goes around the coronal suture, so that's what it is. So it goes like that. And you can see this is the dural fold. You see that? That's the folks cerebri. Okay, And then over here, you can see the superior sagittal sinus, the superior sagittal sinus, venous sinus, right? See here, so the periosteum continues up there, and then the meningeal layer of the juromater comes down, and then it creates that little space. So, in fact, the superior sagittal sinus, that's where most of the blood is, that supplies the brain is drained into. And then this superior sagittal sinus is then joined to the internal jugular vein. You all know, it's my ANB2 guys who are doing the circulatory system. The internal jugular vein. So the internal jugular vein and also the external jugular vein drain the brain. But the external one drains the face and all these other tissues. The internal jugular vein is one that drains the brain. So all the blood from the jurovenous sinuses goes into the internal jugular vein, back to the heart, to the right side of the heart, right? Okay? From there. Okay. Falx cerebri should be on this diagram too. Is it not? It's not. I'm looking for falx cerebri because this is the falx cerebri. See this jural fold right here? That's what is the falx cerebri. Okay? Yeah, so know those jural folds, please, that separate the major structures of the brain, because I will ask about those in the exam, okay? Never mind the textbook. It's being naughty. It should happen. Okay, and so this is the inferior sagittal sinus. Okay, the space that's created on the bottom here, because of the sagittal plane runs there, and there's the superior. And most of it, and the arachnoid granulations, they come from the arachnoid space. Remember, the cerebrospinal fluid is circulating right under the arachnoid, called the subarachnoid space. So the arachnoid grows into this jurovenous sinuses. You see what I'm saying? Especially the one on the top, so they grow into like that. And that's how cerebrospinal fluid is also dumped into the jurovenous sinuses, especially the superior sagittal sinus. Okay? And I'll talk about those arachnoid granulations in a little bit. Okay? So yeah, definitely know those jural folds, those special jural folds. Okay? So there's the superior sagittal, sinus, and there's the venous sinus, jurovenous sinus on the top, and it runs along the sagittal plane. That's why it's called superior sagittal sinus. Right? And this is where the falx cerebri runs, the jural fold. Okay, so now we go to the ventricles. There's 4 ventricles in the brain, and then the way it's arranged is such that so you've got the left and right cerebral hemispheres, so the main parts of the brain, right, cerebral. So what you have is a ventricle in each side of the brain, and it's called the lateral ventricle. Okay, these are lateral ventricles. The one on the left is number 1, and the one on the right is number 2. But we just call them lateral ventricles. Okay, these are the biggest fluid chambers or cerebrospinal fluid chambers in the brain. Okay, in fact, 90 to 80% of the cerebrospinal fluid is produced by the choroid plexuses that are located in here. Okay, so lateral ventricles, those are them, and the lateral ventricles are joined by this connector called interventricular foramina, for plural, foramina. Okay? Okay? Foramen is singular. So interventricular foramina like that. So you've got one on the left, one on the right, and they're joined together so that cerebrospinal fluid flows through all of those. Okay? So, and then this is, of course, an oversimplified diagram. You know what I'm saying? And I'll show you nice ones later on. Okay? So, okay? So 1, 2, 3, 4. So, and this is the central canal of the spinal cord, right? So the third ventricle it follows here from the interventricular foramina. Okay. The interventricular foramina just join those three like that together. Okay. Because another thing that they do in the exam is ask you the flow of cerebrospinal fluid through the brain. Okay. So because that's what it will do, and then it will end up in the subarachnoid space, and then go back to the blood through the dural venous sinus. Okay, so the third ventricle is connected to the fourth ventricle by the cerebral aqueduct. Okay. Oh, so I'm going to use that. That's that. So the cerebral aqueduct, and when we dissect the brain, you will see that because we cut right across the center. So the cerebral aqueduct connects the third ventricle to the fourth ventricle. Okay. Then the fourth ventricle, you've got three little apertures on it. Okay. Apart from the fact that it's also connected to the central canal. So in the spinal cord, there's also cerebrospinal fluid that runs through it. Okay. So that goes through the central canal. Okay. But this one here is got apertures that go to the side like that, and then go to the side, the two lateral apertures, and then the medial one that goes to the trochlear. So it's kind of surrounded like that. If you were to look at it across, you have to see one going that way, one going that way, one going that way. Okay, you see that? So that will be the what connects to the subarachnoid space. Okay, so the ventricle, the cerebrospinal fluid flows from the fourth ventricle into the central canal of the spinal cord, and then it also flows through the three apertures into the subarachnoid space, right? Do you see that now? So where are we starting? We're starting in the lateral ventricles. Lateral ventricles in the cerebrum, right? Those are the big, huge chambers containing cerebrospinal fluid. Then the interventricular foramina joins them to the third ventricle. All right? And the third ventricle runs, remember when we were talking about the thalamus and I said it looks like a yo-yo? And then it's got an interthalamic adhesion that joins the two. Okay? Well, the third ventricle wraps around the middle part of that yo-yo that we're talking about. So the middle part of the thalamus. That's where you have the third ventricle right in there. Okay? And then the third ventricle is then connected to the fourth ventricle by the cerebral aqueduct. All right? And it is this fourth ventricle that is those three apertures that go to the subarachnoid space. And that's how the fluid then circulates into the subarachnoid space. If you want to get a feel of it, 23 mL is in the ventricles of the cerebrospinal fluid. Okay. 117 millimeters is in the subarachnoid space. Okay, and that's what cushions the brain. Okay, so when the cerebrospinal fluid is produced, it was also at the same time being emptied into the dural venous sinuses. At a rate of about 0.4 milliliters per minute. So it's also going to be produced by the ventricles at the same rate, so that the cerebrospinal fluid volume is constant, right? So what happens if these are blocked, or any one of those aqueducts are blocked? Then cerebrospinal fluid starts to accumulate in the head, and you may get hydrocephalus, or and some kids do get it, and then the brain will start to feel some pressure. So they usually go in there and try to correct SSH so that it drains out, because it's constantly being there. All right, produced. All right, so there we go. So I want you to. So the ventricles of the cerebrum, they kind of sort of in like a C-shaped deal, you know, they're kind of sort of like this, and then they're connected and then to the other like that. So if you make a cross section like that, there's going to be a part that you think that it's not continuous, so that's what it's showing here, okay? The lateral ventricles. But they're continuous, they're a continuous space. Okay, I'll show you another picture so you can get an appreciation of it. And then of course you see, remember the thalamus is located right in this space as well. So the third ventricle is located between the lobes of the thalamus, because one is on the right, one is on the left. So inside that, right there by the interthalamic condition. And then the fourth ventricle runs along the brainstem, just behind the or in front of the cerebellum. Okay, fourth ventricle. And then this is the MRI that shows exactly what you see what I'm talking about. If you have this plane XX, so this is what you get. You see how it's not continuous? It doesn't mean that there's separate chambers. It's one chamber, but it's in the form of a C, like so, so that if you take a cut through that, you will see this part, which will be that, and then this part, which will be this. And you will see nothing in the middle and think that it's not a continuous space, but it is a continuous chamber. Okay? So that shows that. And then, of course, the third ventricle, if you're taking a cross section like that, so it runs in the middle. And then this one would have to be lateral, showing the fourth ventricle. Right. Some more MRIs that you can look at, you know, at your pleasure later on. Right. That's showing you. So these are the actual pictures, the actual shape of. You see how I was talking about the lateral ventricles? It looks like a C, and it's actually got an anterior horn and a posterior horn. Okay. So the section we took on that was this. Is it not working? For some reason, my thing is just bit on the anyway. So I guess I have to use this. Sometimes it goes on in the legal like that. I can do that. I'm not sure why it's doing that. I hope it doesn't quit on me. And it's refusing totally. For some reason it switched control to that. It did. Ah, no, please not. Just gonna restart my program? Honestly? What did I do? It deserves this. What side? It just started to pee on me like that. I guess it's too cold too.
Yeah.
Really? You just spit on us? So now I can't do anything until.
Yeah, you gotta wait for it to take.
It's a good thing this is happening. Huh? So maybe I should. It doesn't give me any other option than to wait for program to respond.
No, serious.
Even it's not going to respond to it. Really? This might take several minutes. And you're proud of it? Okay guys, sorry about the technical difficulties, but I hope it's going to work this time. Okay, hopefully we're in good business, right? But anyway, the cross section I was talking about is over here, and therefore you don't see anything in the middle, but you see something over here. But it's a continuous space, as you can see, right? Very good. And the fourth ventricle here is one that is got three apertures that are connected to the subarachnoid space. All right. All right. Very good. So the I hope it's going to work for me. It's working. I hope it saved my other stuff. But the choroid plexus are specialized blood vessels that are surrounded by those neuroglial cells we talked about at the beginning in the central nervous system. What are they called? The neuroglial cells in the central nervous system that produce cerebrospinal fluid. What do we call them? Remember those four? That was four. It was one of them was in the exam. Yeah, ependymal cells. Yeah, ependymal cells. Those ones, they're affiliated because they want to move the cerebrospinal fluid around. But those ones, they are together with the specialized blood vessels that are suspended inside the ventricles, they create cerebrospinal fluid. And the way they do it is that these ependymal cells will pump sodium into the ventricles, right? And then what does water do? Water will follow naturally via diffusion, right? Because water flows from low concentration to high concentration. Okay? So the fluid just comes naturally. So that's how the choroid plexus do. And the answer was right in your face. You're not going to sign it. Okay? So that combination of the ependymal cells plus those specialized blood vessels, they actually look like, you will see when we dissect through the brain next time, they look like cauliflower. They look like tiny reddish cauliflower, and it's going to be suspended inside the ventricle. Okay? And like I was saying, the lateral ventricles produce 80 to 90% of all the cerebrospinal fluid. Okay? They produce 80 to 90%. Did you see how humongous they were? They were so big, those lateral ventricles. Okay? And then, of course, we know how the cerebrospinal fluid moves from the third ventricle to the fourth ventricle via this cerebral aqueduct, right? And then from the lateral ventricles moves into the third via the interventricular foramina, right? Okay, so you should know those spaces and how they are connected up, okay? Then it is from the fourth ventricle that cerebrospinal fluid enters two places. One, the spinal central canal in the spinal cord, and the other is the subarachnoid space surrounding the brain. The one that cushions, that actually does the cushioning of the brain. Okay? Through those three apertures. You got the two lateral apertures on the fourth ventricle and the median aperture that is in the center going up to the, and I'll show you a nice picture with white arrows showing the flow of cerebrospinal fluid, right? Okay, so fourth ventricle to the subarachnoid space. And we know that most of the cerebrospinal fluid is located in the subarachnoid space. Okay, there's only about 23 mils in the ventricles. Okay, so remember what I was talking about? The arachnoid, is outgrowths granulations that grow into the jurovenous sinuses called arachnoid granulations. These are the ones that deposit cerebrospinal fluid into the jurovenous sinuses so that it returns to circulation. Okay, the jurovenous sinuses contain deoxygenated blood, right? After it has been used by the brain. Okay, and then the jurovenous sinuses down into the internal jugular vein. Okay, the internal jugular vein, that's where the cerebrospinal fluid is dumped back into, and then it mixes again with blood at the same rate as the cerebrospinal fluid empties into the jurovenous sinus. It is being produced by the choroid plexus. Okay, and I told you about the rate. It's about 0.4 mLs per minute. Okay, if either too much is being produced and then it's being dumped into the jurovenous sinuses, you get a situation where then cerebrospinal fluid will accumulate in the brain, and that would really not be good. Okay, if it accumulates in the ventricles because one of those cerebral aqueducts are blocked, then its internal And then if it's on the outside, then it's external, that hydrocephalus, the accumulation of fluid, if it's in the subarachnoid space. Okay, and then it will start to put pressure on the brain. Okay, it's almost like when you get a hematoma, if you bump your head and you start to bleed into between the meninges, then that blood will start to squish out the brain. And you could die like that. Somebody actually died, a prominent actor who was married to Liam Neeson. You know, she was skiing in Canada, she hit her head, and then all of a sudden she's like, I'm fine, I'm not going to go to the hospital. So if you ever hit your head against something and you feel slight headache, don't think you're fine. Go get it checked out, because they'll do an MRI and to see if blood is, bleeding into your head, because that could kill her. And she did that, okay? Because she was feeling fine, and she said, no, I'm fine, I'm not going to bother going to the hospital, I just hit my head. And then she died that same day. Because if she had gone to the hospital, they would have seen, the hematoma forming, okay? Anyway, so, gotta know how the cerebrospinal fluid exits into the jurovena sinuses, which is via the arachnoid granulations. All right, and this is the picture of those choroid plexuses. They're suspended inside of these ventricles, and most of those are located in the lateral ventricles, because that's where most of the cerebrospinal fluid is made or synthesized. Okay, by the ependymal cells pumping sodium actively into the ventricles, and then water follows by passive diffusion. Okay, so then you have the cerebrospinal fluid. Okay, so these are secretions from the blood. All right. So how is the blood? How does the blood supply to the head, and how does it work? Okay, so the brain is about 2% the size of the body, right? But it requires a fifth of the circulation of all the blood that is pumped by the heart. Okay? So it requires up to 20% of the cardiovascular circulation. All right? One, because it's got high metabolic rate, right? So the brain will require a lot of oxygen, right, for energy. Two, the brain almost exclusively uses glucose for energy, nothing else. Okay, the brain prefers only to use glucose. So and so the blood glucose is what it works on. It doesn't use any other substrate for energy. Okay. So high metabolic rate, and also the brain only uses glucose to get into for energy, all right? And also the brain has what's called the blood-brain barrier that excludes almost everything else from entering the head or entering the brain, because it could get compromised. Just like also the blood-testis barrier, which is the same thing, that prevents material from affecting the gamete production in the male. Okay, so same thing here. So because of that, you're going to need a lot of blood to go to the brain so that it can, So if you have a stroke episode where a certain blood vessel to the head is blocked, in a few minutes, that brain tissue may die, and that's why people who have a stroke, they kind of have problems controlling whatever part was injured. If it was kind of on the right side, then the left side would kind of off or not working. You know what I'm saying? Because of that. So the brain requires a lot of blood, even though it's only 2% of the. So I want you to be able to explain to me some of those reasons, of course, why that is, why the brain requires so much blood when it's only just 2% of the brain. All right? Okay. So, like I was saying, the blood-brain barrier will surround. Okay. In fact, what is the neuroglial cell that forms the blood-brain barrier? Astrocytes. Yeah. Ependymal make the cerebrospinal fluid. Astrocytes So astrocytes, what did we say they do? They wrap around the blood vessels, and so in a way to anchor the nutrients close to the neurons. So when they do that, when they wrap around these blood vessels, they form tight junctions that prevent things from entering and exiting those capillaries. See what I'm saying? So that's why we said the astrocytes help to form the blood-brain barrier. Blood-brain barrier. Okay. And that's because they form the tight junctions around the capillaries, so that to prevent stuff from entering and exiting. Because the capillaries have big, sometimes the spaces between the endothelial cells lining the capillary can increase, and substances can come out of blood, and then that will be bad. So this blood-brain barrier is very selective. It will only allow the glucose and neurotransmitters as well, like dopamine and them, and so forth and so forth. So it limits what enters the brain for obvious reasons. But you don't want to compromise your center of control unit, the control center. All right. Okay. So gotta be able to tell me the reasons, that the brain requires so much blood for being so small. All right. Okay. So we got blood, that supplies the brain. At the base of the brain, right by the brainstem, there's going to be a circular network of capillaries that will spread blood throughout all the structures like that. Okay. So those circular blood vessels, that's what we're calling circle of Willis, or the cerebral arterial circle. Okay. The cerebral arterial circle starts off, there are vertebral arteries that go up to the head, and they go inside of those vertebral foramina. I think we may have talked about it a little bit earlier on, but I think it's an NP, not you guys. So the vertebral arteries go up, they join to form this basilar artery, which is called basilar because it's at the base of the brain, right by the brainstem. So it runs across like that, and then gives rise to the one in the front here. So I'm going to use green, which is the anterior cerebral artery. Okay, that's one that forms the circular part on the top, and then the one on the bottom will be the posterior, because this is behind, right? Posterior cerebral artery. Okay, and those two join the internal carotids. Okay, the internal carotids, which are these, those are here. Okay, so the internal carotids are bringing blood from the aorta, right, going up, and then there's those two that form a circle like that. The posterior from behind and the anterior cerebral arteries, they join, and then capillaries will spiral throughout under the brain to supply the head. That's what's called the cerebral arterial circle. Sometimes in the brain, I ask you to describe for me the cerebral arterial circle or circle of Willis, and then you tell me that it is formed at the base of the brain by the posterior and anterior cerebral arteries joining the internal carotid artery. Okay? So that it will supply blood throughout the head. Okay? So I need you to be able to describe for me what the cerebral arterial circle is. Okay? That should be in the book normally.
I can't find it.
You can't find the cerebral arterial circle? That diagram should be in there though. Alright? It should be there. Alright? Again, this is showing you the blown up pictures of how the vertebral artery comes in, joins to form the basilar artery, and then the basilar artery, again, this will be the posterior, since it's not written, so posterior cerebral artery. Okay? I mean, it's just a directional name, named because it's in the posterior. And then the one on the top there will be the anterior, right? cerebral artery. And those are the ones that form circle of Willis, anterior and posterior cerebral arteries joining the internal carotid arteries. Okay? Joining the internal carotid arteries. Alright? Like I was saying, from that circle, blood will spiral to the anterior of the brain, to the sides, and to the top, you know what I'm saying? All the way back there. From that kind of circle, and also from the basilar. From the basilar artery, it supplies most of the brainstem, right? You just get branches from it. Alright? This is just showing you the arterial blood supply. And then, of course, the internal jugular veins drains blood from the dural venous sinuses and goes back to the heart. Right? So we know supply of blood to the head, and then supply of blood, drainage of blood from the head via the internal jugular veins, okay, through the dural venous sinuses, especially the one on the top, the sagittal, the superior sagittal sinus, right? So now this is showing you the flow of, cerebrospinal fluid. So we said most of it is produced by the choroid plexuses located in the lateral ventricles. Right. So lateral ventricles, there we have most of the cerebrospinal being produced by the choroid plexuses there. And then interventricular foramina, right, joins to the third ventricle, right? Okay. Interventricular foramina joins to the third ventricle, and you can see this is the direction of blood flow, cerebrospinal fluid flow. Okay. Lateral ventricles, interventricular foramina, third ventricle. Then cerebral aqueduct joins to the fourth ventricle. Right? The fourth ventricle is one that has those three apertures, the two lateral apertures and the one median aperture that joins to the subarachnoid space. Okay, so that's where the blood goes and cushions around the head, the brain. All right. Then also the fourth ventricle is connected to the central canal. And the central canal of the spinal cord is a blind-ended tube, so fluid just goes in and comes back out. Okay, goes back to the fourth ventricle and goes into the subarachnoid space through those three apertures, right? So we are moving, we're going, all right? And then the third part, once the blood is circulated in the cerebrospinal fluid, I'm so used to blood, everything is blood circulating. But once the cerebrospinal fluid enters the subarachnoid space, remember we said the arachnoid grows into the dural venous sinuses that way. You see, this is a blown up picture of these arachnoid granulations that grow through the dural mater, okay, and go into the dural venous sinus. See what I'm saying? And then that's how you dump the cerebrospinal fluid into the dural venous sinus. Okay? So that's next, and I think in your module it was asking what is located in the dural venous sinus, where is cerebrospinal fluid located, and it mentioned ventricles, and then it mentioned venous sinuses, and I think some of you missed the venous sinuses because that's where the venous sinus also cerebrospinal fluid is found. Okay, it's just that it will be mixed with blood on the way back to be recirculated. Okay. So now you can be able to tell me the flow of cerebrospinal fluid, starting with the source, with the choroid plexuses, and ending up in the subarachnoid space, through the and then into the dural venous sinus through the arachnoid granulations. Right? That could be like a 5-point question, telling me those spaces through which the cerebral. Yeah, it's just smiling. Okay. Yep, that's what it is. Because I will do that sometimes, in the exam. I'll ask you to do that. So this is the total flow of cerebrospinal fluid. And thank you so much, ladies and gentlemen. Sorry about those technical difficulties. It only happened once this year, though, this semester. So at least we got spared. And I shall see you in the lab. We'll just meet at 1:45 so that we can finish this and go. Right? Thank you, guys.